Okay, so yes, we've learned a lot in one semester and we're trying to integrate this all together. So with that, I see that I've started with the handout. Oh my goodness. So let's switch over instead to the lecture itself. Much better. All right, plant life histories. So I gave you last time a definition of life histories. I'm going to emphasize this again because I definitely would like you to memorize this. And so a plant life history is a collection of phenotypic traits that then impact fitness via their effects on growth, development, survivorship, and fecundity. And I also gave you another definition that I would like you to remember, and that definition is of fitness. And so remember, fitness is a measure of the reproductive success of a phenotype relative to other competing phenotypes. Okay? And so fitness, it is important to underscore, is a relative measure of the ability to both survive and reproduce. Okay, so we've emphasized that several times during the class. So for today, remember, I have emphasized several aspects of plant life histories. And so plant life histories can differ in terms of how we measure them. And so sometimes we measure life histories, be that the growth rate of a plant, the metabolic rate of a plant, variation in lifespan, survivorship, the population-sex ratio, age of first reproduction, and type of reproduction. These are all examples of plant life histories. Last time I gave you an equation for fitness, and this equation then for fitness will help us characterize and organize plant life histories. So one measure of fitness that we're using here in class is R0. This is the net lifetime reproductive rate. And remember, that's the summation of two things. That's the probability of surviving and also your fecundity, right? And so remember, the summation then of that product of Lx times Mx is our value of R0, our net lifetime reproductive rate of this given phenotype. And so always with fitness, the higher the value, the better. All right, so how can we organize these life histories? And so near the end of last class, I give you this really crappy cartoon of mine. And this cartoon kind of outlines allocation then of carbon into the main plant organs of roots, stems, leaves, but also plant reproductive output, such as flowers, fruits, and seeds, right? And so depending then on how then the plant then can allocate then these resources, here in particular carbon, that then determines future growth rate, but also future allocation. And how then a plant allocates in different environments will also influence its survivorship, okay? So I gave you an equation then for fitness, the sum of Lx times Mx. So here we can see that Mx is then allocation associated with flowers, fruits, and seeds, but then Lx would be associated with how then the plant allocates then to different components of the plant body. And depending on the type of allocation, a plant then can vary in terms of its survivorship. Okay, so here we can see how we can then start to make sense of variation in Lx and Mx in terms of allocation of resources. All right, so the goal then for natural selection is for, within a specific environment, balance various investments in these differing allocations to these different organs in order to maximize fitness. Okay, and so the last slide I gave you at the end of the last lecture was a series of different traits then covered within class. Okay, and this is actually a longer laundry list, but I wanted to make it relatively simple. So hopefully you should recognize several of these different either key words or traits then covered in class. And so I asked you as part of your homework assignment, are these traits positively or negatively influenced then by each other? But then also I asked you to try to go through and see whether or not these traits influence Lx or if they influence Mx. And what you should have noted is that many of these are sometimes negatively associated with each other or positively associated with each other, but also they can have differential impacts on the survivorship and reproductive status then of a plant. Okay, but if I were to boil then down all of these various different traits then listed here, it actually turns out that three traits capture most of the variation in plant life histories. Okay, so if we step back and think of all this variation in plant life histories, actually three traits tend to characterize most of that variation. In particular, the specific leaf area, that is the area of the leaf divided by the mass of the leaf. Remember, that's an indication of allocation to a given leaf, but also variation in photosynthetic rate, how long a leaf lives, and so on. But also how big a plant is. Plant height turns out to capture a lot of variation in plant life histories. And also another trait, seed size. Okay, so specific leaf area, plant height, and seed size capture most of the variation in plant life histories. So I would like to introduce again this notion of a trade-off. We've used trade-off as a term throughout the class, but let's just give a kind of more of a colloquial definition. And that is a trade-off is a balancing of factors, all of which are not attainable at the same time. Another way of thinking of it is that a trade-off is just giving up one thing in return for another. So within biology, we see quite a few trade-offs. In fact, I'm going to give you a laundry list of several trade-offs that we observe within biology. So I would like to ask and, well, basically pose a question then to you. Why do we see trade-offs in biology? Okay. So if you can kind of note that off to the side, we're going to return to this, but this would also make a very good final exam question as well. Why do we see trade-offs within biology? Okay, so let's switch gears a little bit, but let's zoom in on one aspect of variation in life histories, but also something then that influences variation in survivorship, but also in reproduction. And let's zoom in on one specific aspect of a trade-off, and that is something associated with the defense strategies of plants, but in particular allocation to defense. So a core dilemma of plants is associated with whether or not natural selection will select then for increased allocation to defense, but that allocation to defense also diverts resources away from production of new leaf area, and because of which future growth rate is also limited. So if a plant then allocates more resources to defense, it's thus then going to limit potential future growth rate. But also, it's going to diminish its capacity for maintenance, storage, and also reproduction. Okay? So here's a dilemma then of plants. Should I grow or should I defend myself? Should I allocate my limited resources to defense, or am I going to allocate those resources to growth? A trade-off. This is a dilemma of plants. So let's give one example of such a trade-off or a dilemma. So I'm asking here, what is the cost of defense? And so I'm listing here several primary compounds, such as carbohydrates, organic acids, lipids, nucleotides, amino acids. These are compounds associated with growth rate. Here are compounds associated with secondary chemistry or secondary compounds, such as terpenoids, phenolics, alkaloids, other nasty nitrogen compounds. Remember, these are the really toxic compounds associated with defense in plants. And what I'm showing you here is a unit of comparison. This is the cost of producing either primary or secondary compounds. This is the grams of glucose required to produce a gram of compound. So if you notice, it takes about 1.07 grams of glucose in order then to produce a gram of carbohydrate. Okay? The cost is a little less for organic acids, a little bit more expensive for lipids, but around the same for about nucleotides and amino acids. But now notice how much it costs to actually produce secondary compounds. Secondary compounds are costly. It costs more grams of glucose to produce a gram of terpenoid, phenolic, and alkaloid, or other nasty nitrogen compounds. Okay? So then the cost then of defending yourself is the cost of loss of fitness to committing resources then to growth rate, but then also a cost is the loss of fitness to committing resources then to defense. Okay? So allocation to defense can increase your survivorship, but it then comes at a cost. That is, you won't be able to make as many primary compounds necessary in order to grow, okay? Or to allocate to reproduction, a trade-off. So allocation to reproduction. So should a plant then allocate to growth or reproduction? So here are the typical scenarios that natural selection then grapples with depending on the local environment. How much to allocate to reproduction, as in flowers, fruits, and seeds? How many seeds then should selection then influence a plant to make? How much to spend on seed quality? So we can define another aspect of this trade-off aspect of growth or reproduction by a measure known as reproductive allocation. Reproductive allocation is the net resource investment in reproduction that's diverted from vegetative investment to reproductive investment. And it's simply a proportion or a percentage. That's how much of the total allocation is then spent on reproduction versus vegetative investment. All right, so what we're going to then do next is focus on differences in allocation to reproduction. And that gets us to a fun topic, and that is variation in allocation to seeds. In particular, we want to focus a little bit more on seed size. And so here I'm showing you a picture. This is a typical example, if you walk along at least a tropical beach, all the various different seeds that you'll likely find washed up on the beach. They take different sizes, but also different forms. And so there's some small seeds, but also much larger seeds as well. And you can study the diversity of plants mainly by studying the diversity of different seed forms and sizes. So seeds in general are a care package from mom. And so the female component here on a plant, obviously the reproductive structures on a plant, but the seed itself really is a care package for the future generation. And so seeds are a care package from mom. So if you think about your loving mother allocating differently than to different children, and so you can think of a big seed is a very fortunate offspring, and because of which that fortunate offspring, because of the high investment from mom, will have a higher probability of surviving, especially if the world is filled with, is marked by limited resources and also intense competition for those limiting resources. On the other side of the coin, a small seed represents less allocation to future generations. And so this would then mark a less fortunate offspring with a lower probability of survivorship. And so mom then is taking her hard-earned resources and then allocating differently than to the future generation. And you can see that by the size of a seed. So a big seed, fortunate offspring; small seed, less fortunate offspring. So let's give a quick tour of the range of variation in seed sizes in the plant world. And actually this is pretty cool because the smallest seed known comes from an orchid. And so here's a forest orchid here. And so this species then of orchid, the seed is only about 0.1 to 0.2 millimeters in diameter. And so here is an actual orchid seed, and this is actually one you can see. This is a little bit larger. This one's 1 millimeter, and so it's substantially larger than to this one species where the seed size is about 0.1 to 0.2 millimeters in diameter. Incredibly small. Very little allocation of carbohydrates then to the developing zygote. And this seed size, in fact, it's so small, it's about the size of a speck of dust. In fact, I've seen these seeds from the species of orchid, incredibly hard to actually see it with the naked eye. So, quite small. So what's the opposite side of the spectrum? So if we want to find the largest seed on the planet, you actually have to go to the Seychelles Island. It's actually a group of islands off the coast of Africa and Madagascar. Beautiful location. And within this endemic palm, you'll actually find, within this endemic species of the Seychelles, the largest seed in the world. It's called the Coco de Mar, or the double coconut. So it's a palm, and it's actually not in the same genus as the coconut. It's actually in a completely different genus. But this seed can weigh as much as 40 pounds. So literally this thing, if it fell out of the tree, could kill you. Always love going to Google Images. Here's a happy honeymooning couple finding for the first time a true version of the Coco de Mer, and it's the largest seed. So an incredible range of variation in maternal allocation to the future generation. What's interesting about seed size is that it varies enormously depending on where on the planet you go, but also the different environment in which you study. And so here what I'm showing you is we're plotting seed mass, and this is actually on a log to the base 10 scale versus the frequency of different species with a different seed size. And so this is in milligrams here. And so what you'll actually find is that there's, gosh, well over almost 1, 2, 3, 4, 5, 6, 7, well over 7 orders of magnitude of variation in these different locations, ranging from a field outside of Sydney, Australia, to a field outside of Sheffield, England. Here is a dune area within the state of Indiana. There's the Californian chaparral. Notice the enormous variation in seed size then that we see. And so realize that this variation in seed size is variation in maternal investment to the future generation. So these are the maternal side then of allocation. So these then are moms allocating their hard-won carbon in then to these care packages known as seeds that then will influence the growth and survivorship of the future generation. But notice that seed size varies enormously. So why is that? Why does seed size vary so much? Again, a great question for the final exam. A little bit of phylogenetic background. It actually turns out here is a phylogeny of the angiosperms, and so this is a simplified phylogeny of the angiosperms. You see the monocots here, the roses and the asterids. And we're plotting then here, if you see these red or these kind of larger or smaller circle areas, these are the location of the top 20 divergences in seed size. So these then are taxa across then the angiosperm phylogeny, where we see huge variation in seed size, ranging from some of the smallest seeds to also some of the largest seeds. And so if you notice that this variation in seed size occurs quite frequently across the angiosperm phylogeny. And so again, these are taxa with very large and very small seeds, and this has evolved multiple times just within the angiosperms. So within each of these different lineages, we see strong selection for small seeds or strong selection for very large seeds. So again, I want to ask you then this question, and again, this question will appear on the final exam. Why then do we see strong selection for either small seed size or very large seed size? And again, we see this over and over again with strong selection for either small or large seed size. Okay, but we have to remember that all of this allocation is done under the context of trade-offs, okay? And so here what I'm showing you is an interspecific relationship. And when I say interspecific, that means that each data point here is for a different plant species. And so this is a comparative relationship across species. And we're plotting seed mass versus the number of seeds produced per canopy area of forest per year. If you notice, there's a negative relationship. And that is when we find then a forest where we have a lot of species dominated by species that produce big seeds, they tend to produce fewer numbers of seeds per canopy area per year. Whereas if we find ourselves in a forest then that's dominated by species that produce small seeds, they tend to produce a heck of a lot of small seeds per unit area. Notice this is a logarithmic scale. So these species are producing, gosh, orders of magnitude more seeds than these species then that produce large seeds. Notice that the slope here is actually -1. So does -1 tell you anything special about the fact that this is not only a negative relationship, but it is also a trade-off. Okay, and so we will come back to that in a little bit. A negative relationship indicating a trade-off. Here then is another similar trade-off, but this is between seed quantity and the quality of the seedling growth rate. So here is the seed, we're plotting seed size. So this is the mass then of a seed versus RGR. This is the relative growth rate then of a plant. And notice within each one of these separate lines, these are effectively intra and interspecific relationships. And so within each of these separate lines, this then is connecting closely related species then within a genus, within each one of these lines. But then also if we notice across all of these different data points together, we're now looking across all of these different species. And so in general, we see mostly a negative relationship between seedling growth rate and the reserve status then of a plant. So in general, what that means is that as then selection selects for a larger seed size, that comes at a cost of allocation then to growth rate, and hence a negative relationship or a trade-off between how fast a plant can grow, that is allocation to growth, versus allocation to reproduction. Now notice that sometimes within then closely related species, we don't see a negative relationship. And this indicates that potentially there are other factors at play outside of this trade-off, this univariate trade-off between seed mass and relative growth rate. Okay. Now, we note here that relative seedling growth rate decreases with seed size, but seedlings from large seeds have higher survivorship. Remember survivorship? This is LX. So again, step back. What we're doing here is I'm giving you several different examples then that differences in allocation can influence not only growth rate, but also can influence survivorship. So here's a case where allocation then to seeds then will decrease your growth rate, but yet seedlings then with higher allocation and a greater seed mass will have a higher survivorship, right? And so this is the loving investment then from mom. So as moms then invest more in then seeds, seedlings then utilize then the resources to better eke out an existence in an environment where resources are limiting so that they actually have higher survivorship. So why don't all plants have large seeds and high survivorship? And for that matter, high fecundity. So how come all the plants that we see outside don't have huge seeds? Why is it that we see such variation in seed size? But similarly, why don't we find plants out there packed then full of defensive secondary chemistry? So why can't plants have huge seeds and huge numbers of secondary compounds so that you basically design by natural selection the perfect plant that's well defended and well allocated for the future generations? Okay, so we have to talk a little bit about optimal life histories. And optimal life histories help make sense of this dilemma. That is, how come all plants don't have large seeds? And this reflects the fact that there's a fundamental trade-off between fecundity and survivorship. So remember in our equation then for fitness, or R sub naught, that then equals the summation of Lx times Mx, it turns out that there's a fundamental and deeper trade-off between Lx and Mx, that is, between survivorship and fecundity. And so here what I'm showing you is actually a theoretical plot showing and predicting that if we plot out a measure of the survivorship then of a given phenotype, or Lx, versus its current fecundity, or Mx, in general, there is a negative relationship between Lx and Mx. Effectively, if R naught then, if fitness then is maximized, or is, in this case, 1, then the summation then of these probabilities then can be changed back and forth, but you cannot exceed then a maximum fitness value then of 1, indicating then that Lx and Mx actually trade off against each other. And so species in general with low current fecundities, so if you don't produce a lot of babies or a lot of seeds, you tend to have a high survivorship. On the flip side of the coin, species with high current fecundity, those then with high birth rates, with high allocation then to seeds, have low survivorship. And in general, this tends to be a pretty good rule or a law across all of biology. In fact, if you step back and look at the diversity of life, we see this fundamental trade-off between fecundity and survivorship. You just can't do both at the same time. So why is that? Why then are fecundity and survivorship negatively associated with each other? Again, another great question for the final exam. Okay. So let's then try to sum up a little bit about a theory of allocation. So a major assumption then that goes in to our understanding of this negative association between fecundity and survivorship, but in particular in terms of linking that then to allocation patterns and differences in biodiversity according then to what we see in differences in allocation, is that ultimately resources are in finite supply. And in general, these resources then have to be distributed or allocated amongst different chores, that is, growth, maintenance or defense, and ultimately reproduction. And so why then do we keep focusing back on allocation? Well, allocation then of resources, allocation of carbon, allocation of nitrogen, is key then to linking life history theory, growth rate, and physiology, but also all of these different traits that we've been measuring in a unified kind of context. Okay. So a key then to life history theory is that resources are allocated among competing functions. Allocation to leaves then competes then to allocation to reproduction. And this necessarily generates trade-offs, right? These are zero-sum. Remember, we talked at the beginning of class in the first few lectures about zero-sum trade-offs. This is a fundamental trade-off because resources are allocated among competing functions. Now, sometimes this is not entirely true. Sometimes we find fruit that are actually green and photosynthetic, so that fruits can help in some way augment the cost then of their production. Stems can also act as biomechanical support. So instead of thinking of a stem as an allocation that is a detriment to future reproduction, well, also stems provide important biomechanical support that can help ensure and maintain that you have reproduction into the future. But also in terms of reproduction, sometimes reproduction is vegetative, so that we don't have to allocate then to flowers or even to to pollinators or even to seed dispersers. Sometimes if we do asexual reproduction, we can kind of get away with the cost of reproduction. So there are some exceptions. Okay, so if we step back and kind of look at the grand diversity of plants out there on the planet, we can organize then the diversity of plants along then different trade-off axes. And these trade-off axes, again, reflect the fact that allocation to one aspect of plant biology comes then as a trade-off to another aspect of plant biology. And so this is a long laundry list. But in general, we find that plants then of the world can either be good colonizers or they can be good competitors. So you can allocate then to colonization, that is increased allocation then for more reproductive output, more seeds, more asexual reproduction in order to send out tillers, to send out various aspects of reproduction into the landscape. Or you can be a really good competitor. That is, you can allocate all of those resources to your stems or your roots, and you can outcompete then your neighbor instead of allocating in terms of colonizing new habitats. So plants are either really good at colonizing new habitats or they're really good competitors. Similarly, you can allocate against herbivory or you can allocate more toward competition. Another trade-off is, well, you can allocate more to roots or you can allocate more to shoots. So plants then that are really good at competing then for light tend to allocate more to their shoots. plants then that are really good at allocating in order then for sucking up water and nutrients, allocate more to roots. Also, in terms of reproduction, there tends to be a trade-off between dormancy and dispersal, so that those seeds that are good at dispersing tend not to be really good at waiting out long periods of time in dormancy. But also, there's a fundamental trade-off in terms of longevity and growth rate. So in general, if you tend to grow very fast, you tend not to live very long. Similarly, survivorship and reproduction is often traded off. And we've emphasized a few times defense and growth rate trades off against each other, but also growth and reproduction. And the list goes on and on. So these are all fundamental trade-off axes that you can characterize the diversity of plants according to. So how can we organize then all of these different life histories then together? So let's step back and talk a little bit more about the evolution of plant life histories. And I'm going to bring up a topic that we discussed near the beginning of class. And so when we were building our ecological and evolutionary lexicon, I introduced to you the concept of a fitness landscape. And this is coming from the earlier work of Sewall Wright. And so remember, the fitness landscape, we're just plotting in 2 dimensions here, all the various different combinations of different genotypes together. And so recombining all the different genes then that's available for recombination. And then we're plotting out on this little heat map or this 3D map, variation in the fitness then of those different genotypic recombinations. And so remember, this is a fitness landscape is a map then of all possible genetic combinations in the population. And this is the variation then that natural selection will operate on. And in general, selection is going to eliminate the least fit genotypes in valleys and then favor the most fit genotypes then on the hills. And evolution then will always push populations upward, up hills, up until the adaptive peaks. Okay, and so the analogy here is that evolution then is a walk on a fitness landscape, and evolution will always push then a population uphill to the most favorable genotype or phenotype. So remember, this is a powerful metaphor. And so the quote is that a central problem of evolution is that of a mechanism by which species may continuously find its way from lower to higher peaks. And so the problem then with thinking about fitness landscape is how then do we define all the possible phenotypes or genotypes? And in terms of fitness trade-offs, evolution by natural selection will then act then on this life history and ultimately fitness landscape. And the initial conditions then here is going to then set the stage for how then a population will then move across this landscape. But also, how then do we characterize variation of fitness landscapes in both space and time? So in short, how then can we use a fitness landscape to help us understand variation in life histories? So remember, selection will act on optimal life histories so that it will maximize fitness in the face of many of these different trade-offs. And so this is the problem. How then can natural selection optimize then a given life history in the face of all of these different trade-offs? Okay. So I would like to introduce to you a really nice example that was brought about by a professor at Cornell University. He actually did this work in the 1980s. And what Carl did, and he also wrote one of the books that you're using for class, was that he was interested in reconciling this problem. And he wanted to use plant branching patterns to illustrate how differing selective pressures has ultimately led to the diversification of land-plant diversity, or the diversity of land-plant phenotypes. So if you remember, again, back to, about midway through the class, this is a nice kind of summary diagram outlining all of the different morphologies and anatomies and structures then that we've learned then throughout the class And so you'll recognize stomates, and gosh, we have a trachid here. We have some branching architectures. We have different organization of secondary growth. And we also have, oh my goodness, some interesting reproductive structures, either with one or two integuments and leaves then themselves. And so we can place then all of these important synapomorphies associated with their important selective then pressures as a function then of time, then through the geological time periods. And so this figure then kind of encapsulates everything that we've learned about the evolution of diversity within the land plants in terms of the key synapomorphies. And so what Carl Nicholas was interested in doing was stepping back and thinking about the earliest land plants, in particular the earliest land plant morphology. And he asked the very simple question, well, how then can we take such a simple plant, in particular, if you recognize this, this is Cooksonia, the first land plant fossil. How can we take such a simple, humble beginnings and evolve from such a simple, humble beginnings, ultimately all of the land plant diversity that you see outside in the world? And so what Carl wanted to do was use this fitness landscape to understand land plant evolution and the evolution of plant diversity, but through the lens of these first defining traits of land plants. And actually, Carl was one of the kind of pioneers to use computer simulations in biology. And in fact, in the early 1980s, he tried to scrounge up as much computer power as he could to simulate the evolution of land plants on a computer. Basically, can we take everything that we've learned in class, but can we start with a few simple rules at the beginning and let the computer evolve the amazing diversity that we actually see outside in the world today? And he started by defining then the first phenotypes then of plants. And he did so by defining 3 different key traits. And he started with the simple branching architecture of Cooksonia, and he defined then this branching architecture by a bifurcation angle, the angle by which that plant is then going to be branching, but also a rotation angle. And that's basically whether or not the branch is coming out toward you or back behind the wall, and also a different set of probabilities then for as the plant grows for making a new branch. So we wanted then to create a very simple plant characterized by simple rules of differences in branching angle, the rotation of the angle, but also the probability of branching. And so with that, he created a fitness landscape defined not by genotype, but instead by the frequency of branching, from sparse branching to dense branching, differences in the rotation angle, and this would give you a really bushy plant or a very erect or planted plant, and differences in the bifurcation angle, again going from an erect plant to a more splayed out plant. This was, from a computer point of view, the phenotypic space. Okay? And remember, this ultimately is going to be linked to the genotypic space. So can we simulate the diversity of plants on fitness landscapes? And so the next thing then that Carl did was define then the components of fitness to a plant. And so if you take everything that we've learned within class, we can characterize the different components of fitness in terms of how well a plant can intercept light. Remember, light then is important then for obtaining ultimately carbon and for photosynthesis, but also differences in mechanical stability, differences in your ability to withstand wind and gravity, but also fitness is going to be influenced then by reproduction. But also fitness is going to be influenced by water loss. So notice that light interception, mechanical stability, and water loss, that's going to be influencing your survivorship. Whereas reproduction, allocation or reproduction, is going to be influencing then, obviously, your reproductive output. So here we have the basic components of LX and MX. Light interception, mechanical stability, reproduction, and water loss. So depending on the environment, selection will then act to either maximize or minimize these various different components. So again, think in terms of R sub naught and the differences in the environment and how then natural selection will then try to maximize fitness. So the initial conditions then of this computer simulation was actually something that looked very Cooksonia-like, this very simple branching architecture. And remember, these plants were only a few centimeters tall. These were dinky, really diminutive plants. So let's start then with how this computer algorithm works. And this computer algorithm works by setting up a series of adaptive walks. And so here he starts at an initial condition defined by the frequency of branching and the rotation angle and also the bifurcation angle. Basically, this is what characterizes the first Cooksonia species that we see within then the fossil record. Now, how this computer algorithm works is that you start at the initial condition with your traits of branching, your rotation angles, and your bifurcation angles. But then, depending then on the environment, you're going to search the immediate neighborhood. So you can think of this as genetic recombination. And we're going to then move to a new phenotypic space if then that plant is fitter than the previous plant. That is more fit than your initial starting condition. Then we're going to repeat this at each time step over and over again. And depending then on the environment, you'll then see phenotypes maybe explore a wider search area if then fitness continues then to increase as we walk then along the fitness landscape. Now it could be that there are alternative phenotypes as well. Now a phenotype then will stop exploring then this space if then it reaches what? If it reaches an adaptive peak, right? If that then combination then of traits is the most fit compared to everything surrounding in the neighborhood. Okay. So here are some examples of what these adaptive walks actually look like. If we start then from a Cooksonia-like original ancestor, but then we select then for different plants that maximize dispersal or maximize light interception or maximize biomechanical stability. And so if we take that initial Cooksonia design and we select then for a phenotype that maximizes light availability, what you actually end up is a phenotypic walk through phenotypic space that results in plant branching patterns that look like this in example B. If you notice, maximizing light availability gives you these very broad or splayed out canopies that for the size of the plant actually maximize light interception. Alternatively, if then natural selection maximizes dispersal ability, you want to put all of that allocation into a really tall and minimal branching architecture. And if dispersal is by ballistic dispersal, that is seeds being thrown from the top of the plant or spores then being dispersed by wind from the top of the plant, the taller you are, the greater your dispersal availability, and because of which that will select for a certain phenotype. Alternatively, biomechanical stability will then select for different phenotypes that then closely abide by not only the Euler buckling equation, but will minimize the probability of buckling due to gravity. And you'll get a different set of branching patterns. So what's really interesting is that depending then on how you select by either dispersal, light, or stability, you can end up with different branching patterns that look quite different from the initial Cooksonia design. So here then are the different phenotypes, and for the different adaptive peaks, if you select for just one task. That is, if you select on Cooksonia for maximum spore dispersal, here's the resulting phenotype. If we select for maximum light interception, here's the resulting phenotypes. If we select for minimum surface areas, we actually select for something that remains very Cooksonia-like, a very diminutive small plant with very little branching. And here's selection for maximizes mechanical stability. So from a very humble initial beginning, depending on how selection then acts, we can then end up with different phenotypes. Now, what's interesting is look what happens if we then select for two different tasks. Instead of just selecting for maximum light capture, what if we select for maximum light capture and dispersal, or maximum light capture and biomechanical stability? Well, it turns out here then are the different phenotypes. And notice that they're different than if you select for just one task. So here what I'm showing you here, MDLM, that corresponds then with your key down below. Here then is the phenotype that maximizes two tasks. This is a plant that's good at biomechanical stability and dispersal. Whereas in B, here then is these are phenotypes that are good at maximum mechanical stability, but also minimizing external surface areas. Here then are phenotypes that maximize the two tasks, light interception and mechanical stability, and so on. And so you can see by the different combinations, LS, DS, and DL, we get additional types of phenotypes. Okay, so I gave you examples of adaptive peaks for one task, adaptive peaks for two tasks, but look what happens if now we ask the computer to maximize three different tasks. These then are phenotypes then that emerge after an evolutionary walk, an adaptive walk through this adaptive landscape for maximizing, in this case, ML and D, mechanical stability, light interception, and dispersal, or D down here, LD and S, light interception, maximum spore dispersal, and minimizing surface areas. And if you'll notice, we start developing a huge variation in different morphological and different phenotypes from, again, a very simple, humble beginning with just the Cooksonia simple branching architectures. And I think very quickly you'll start to see, oh my goodness, these different plant branches seem then to correspond with things that I've seen in the real world. Now, you can also ask the computer to maximize each of these different tasks. And what you'll notice is that all of these phenotypes actually have optimal designs that either maximize or minimize each of these different characteristics. So we've gone through selection for adaptive peaks for one task, adaptive peaks for two tasks, adaptive peaks for three tasks, and adaptive peaks for 4 tasks. So notice what happens as we select for more tasks and optima. We have highly variable morphologies. Now we can take this example of this very simple computer model, this Nicholas example, showing how simple branching patterns and selection for, depending on the environment, for either 1, 2, or maybe even 4 or more tasks can lead to an enormous diversity then of phenotypes. And in general, land plant evolution has explored this phenotypic branching space due to differing selective pressures to maximize fitness within differing environments. And again, these highly variable morphologies, oh my goodness, we've seen some of these things. We've seen some of these rhinophytes, the Zostrophyllophytes, these various different Trimerophytes, these progymnosperms, conifers, and even angiosperm branching morphologies. So from a very simple computer simulation with a very few rules, we can simulate an adaptive landscape and the adaptive walks then through this adaptive landscape. But that adaptive landscape is then defined by then these differing trade-offs and selection in different environments to then maximize or minimize different types of trade-offs. So in terms of fitness landscapes, the more then peaks then that we have, that is, the more options natural selection optimizes, the lower than the peaks. So here we went from basically optimizing one task to multiple tasks to more tasks, we see a change not only in the relative fitness, but also we go to more complex fitness landscapes. And because of which, as we get more complex fitness landscapes, and as selection then maximizes or minimizes different components, light interception, dispersal, different aspects of survivorship, such as water loss, and so on, we now get many more fitness peaks, indicating that we have many more opportunities for biodiversity. And so biodiversity in many ways is reflecting this complex fitness landscape. So back to biology. Okay, so again, how do organismal lineages search then this phenotypic or genotypic space? Well, remember, this is done by mutation, recombination, polyploidy, hybridization, but also in terms of plasticity. And we talked also near the end of class about phenotypic plasticity. Now, what limits then the search in terms of how much variability then we see within populations? Well, that has to do with differences in terms of development, limitations in terms of how much you can actually recombine your genome, genetic impediments such as linkage or epistasis, selection. Okay, you can actually have very strong stabilizing selection in order then to limit then searches, but also drift in the flat landscapes, and that's going to be influences by the strength then of different trade-offs. So we can bring then biology back into this. Okay, so let's now wrap all of this up. So I would like you to consider the following. What are the ecological and evolutionary ramifications of very complex fitness landscapes in the context of large numbers of physiological and life history trade-offs? So with that, I would like you to ponder that as we then move in then to the last part of the lecture here today. So here again, I want to remind you of the major adaptations to land and air as selective pressures then have pushed then these aquatic green algae and out then of the very warm and humbling nature of the oceans and the seas onto land. And in terms of a summary, more complex selection, more complex and more trade-offs, I should say. The result is that we have more adaptive peaks on these landscapes, but the peaks are lower, and because of which the fitness differences then between species are not as great. And the take-home message for this is that the more trade-offs then that are introduced in optimizing fitness, the more optimal peaks then there are on the landscapes. That is, the more opportunities we would expect then to see for increased numbers of species. So there are more ways of making a living and more potential diversity. So the more trade-offs that are included in optimizing fitness, the more ways of making a living and the more potential diversity then that we see. Okay, so how does this relate to stabilizing versus diversifying selection? I would like you to go back and review then your notes for this, but also I would like you to think about what this means about how we think about how different environments then differ in terms of the diversity of plants then that we see in those environments. Okay, so with that, I would like you to leave you with several of these different questions that you should be able then to answer at the end of this. And with that, I will sign off and we'll see you on Thursday. And with that, thank you very much. See you soon. Bye-bye.